While many countries battle a difficult second wave of the COVID-19 pandemic, a light at the end of the tunnel has been offered by the rapid approval and rollout of several new vaccines. Previously, it has taken many years to develop and bring to market new vaccines. But with a global emergency, large amounts of funding and willing trial participants, scientists have been able to offer a range of vaccines in just a year. Many of these are pioneering next-generation technology, which can be rapidly scaled up to meet global demand. To start, we can look at how vaccines are traditionally produced. Classically, eggs are used to cultivate viruses. It's a method that has been applied for over 70 years and is used to develop either live attenuated or inactivated vaccines. Live attenuated vaccines use a weakened form of the virus, where the capacity to cause disease is minimized or the ability to replicate is prevented. Inactivated vaccines are killed by applying either heat, acid, or radiation. The weakened or killed virus is then injected into people, activating the body's immune response to provide protection against disease. For the virus SARS-CoV-2 and its disease COVID-19, there are currently a few inactivated vaccines being used. Two examples are the Sinovac vaccine, approved for emergency use in China and Brazil, and Bharat Biotech vaccine in India. This is still the most common approach to developing vaccines. But there are now 4 other types of vaccines being used against SARS-CoV-2. Genetic, viral vector, protein subunit, and virus-like particles. The majority target the exact same part of the virus. This is the spike-like structure or spike protein on the surface of the virus, which binds to human cells and allows the genetic code to enter and infect the cell. How each type of vaccine targets the spike protein differs. The most revolutionary are genetic and viral vector vaccines. Genetic vaccines inject DNA or mRNA code for just the spike protein directly into the body. Both Moderna and Pfizer-BioNTech use this method and were some of the first vaccines to receive widespread regulatory approval for emergency use. To protect the genetic code, a lipid nanoparticle coat surrounds the mRNA. This unstable molecular protection is the reason why the vaccine needs to be kept at extremely cold temperatures. After injection, the lipid coat fuses with a cell, allowing the mRNA to enter the cytoplasm and start coding for the spike protein. Both the spike protein and spike fragments protrude from the cell. When the cell dies, these spike proteins trigger the immune system to provide protection. But we'll come back to this later. Viral vector vaccines use another virus as a vehicle or vector for carrying the code into the body. It's often an adenovirus, a common virus that causes mild cold or flu. As it's more stable than a lipid coat, viral vector vaccines can be kept at refrigerated temperatures. The Oxford AstraZeneca vaccine uses a chimpanzee adenovirus that has previously been shown to have no effect on humans. It contains the modified RNA spike sequence for SARS-CoV-2, but translated into the DNA code of the adenovirus. The adenovirus vector vaccine is injected into the body. When it reaches a cell, the genetic code is released and makes its way to the cell nucleus. An mRNA strand is produced, which is ejected into the cytoplasm, where it is read and coded into SARS-CoV-2 spike proteins that fill the cell and protrude from the exterior. When the cell dies, releasing the spike proteins and fragments, it triggers the immune system. The immune system contains antigen-presenting cells which can engulf loose spike proteins from a dead cell and present the fragments on its surface. Another vital immune cell, a T helper cell, detects the spike protein fragments on the antigen-presenting cell and activates. At the same time, another immune system cell, a B cell, also detects the spike protein from the vaccine or dead cell, engulfing it to display the spike fragments on its surface. When an activated T helper cell reaches a B cell with the spike protein fragment on its surface, it activates the B cell, causing it to produce antibodies. Antibodies are proteins that can attach themselves to the spike protein. If someone is exposed to the SARS-CoV-2 virus, antibodies bind to the virus spikes, blocking it from infecting a cell and preventing COVID-19 disease. It's worth noting that the immune system is a very complex topic, simply expressed here to show how a combination of many cells and systems dictate the strength and length of vaccine protection or immunity. Although they've been in development for decades, this is the first time both viral vector and genetic vaccines have been approved for use in people, and marks a turning point for the future of other new vaccines and treatment options. To ensure they're safe and effective, all vaccines undergo a strict clinical trial process, from preclinical to phase 1, 2 and finally 3. To speed up the trial process for the COVID pandemic, some of these phases are run concurrently. All the clinical data is gathered and submitted to regulatory bodies, who decide whether a vaccine is approved based on safety and efficacy. The efficacy percentage, which is often cited, is a particular calculation based on the number of volunteers who test positive for COVID-19 and not on the number of people out of 100 who are protected by the vaccine. In the case of Pfizer-BioNTech, 43,661 volunteers were recruited to the trial and split into two groups, vaccine and placebo. 170 people in the trial tested positive for COVID-19. Of those, 8 had received the vaccine and 162 had received a placebo. Researchers calculate the fraction of those who got sick in both the vaccine and placebo group. They mathematically find the difference between the two fractions and determine the efficacy percentage. If the fractions are the same, it would be 0%. In the case of Pfizer, they calculated it as 95%. It's a good indication of how well a vaccine works, but not that 95 out of 100 people are protected. Real-world effectiveness, complicated by many factors, can be very different. It's not always the first vaccines to market that are the best. Many other vaccines are still going through clinical trials and could provide stronger or longer immunity, have better safety profile, or be cheaper and easier to produce and distribute. There are 2 other types of vaccines not yet discussed that are in clinical trials for SARS-CoV-2, virus-like particles and protein subunits. Virus-like particles, or VLPs, are currently used for a handful of other vaccines. They work because of their resemblance to the virus, but contain no core genetic material that causes disease. On their surface are exact replicas of the virus spike proteins, which, when injected, will trigger the body's immune response. For SARS-CoV-2, Canada-based Medicago has developed a plant-based VLP currently in phase 2 clinical trials. Protein subunit vaccines are already used for a few viruses, such as the human papillomavirus or HPV, where some strains can cause cervical cancer. The spike proteins of the virus are grown and harvested. These can either be injected or constructed into nanoparticles and given as a vaccine. For SARS-CoV-2, Maryland-based Novavax has developed a nanoparticle protein subunit vaccine that is currently in phase 3 clinical trials. The speed at which these vaccines have not only been developed and approved, but also produced on a massive scale is a remarkable feat of science. As the pandemic shifts gear with the arrival of new variants, there is some concern that this could impact the effectiveness of vaccines. But there is hope. These new vaccine methods are based around the genomic sequencing of the virus, utilising its genetic code to produce the spike protein. This means that with new variants, the code can be updated, whilst the processes remain the same, meaning new updated vaccines can be developed and produced rapidly, helping us keep one step ahead of the global pandemic.